Hi everyone. The topic assigned to our group is called sulfonamide antibiotic resistance. So, before we go into the nitty-gritty of this presentation, I would like to start off giving the definition of sulfonamide. Sulfonamides are also known as sulfur drugs. They are antibiotics that inhibit the biosynthesis of folic acid, and these folic acids are needed for bacterial growth. So basically, they inhibit the growth of bacteria that need folic acid to live. Sulfonamide targets the enzyme dihydropteroate synthase, DHPS. They inhibit them in the folic acid pathway. And DHPS plays an essential role in making folic acid. And since the bacteria we're trying to reduce need folic acid to grow, we're using sulfonamide to inhibit the essential enzyme that creates folic acid. On the left is the structure of sulfonamide, and examples of sulfonamide includes sulfur methoxazole, sulfur diazine, sulfacetamide, and sulfioxazole. You might be asking, why does this topic matter? I would like to talk about the importance of sulfonamide. Sulfonamides are used to reduce bacterial infections. They inhibit the bacterial enzyme that was mentioned earlier, DHPS, dihydropteroate synthase. They can be used to treat type 2 diabetes and melanoma. They can be used to treat skin conditions and inflammatory diseases. Now, let's go back to why sulfonamide resistance is important. Okay, so let me try to paint this picture. We're humans. We have bacteria in our body. We have good bacteria and we have bad bacteria. And as we all know, harmful bacteria are not good for our body. It's not good for our body system at all. And that is where sulfonamide comes in. Sulfonamide comes in as an antibiotic. We take that antibiotic that stops those bacteria from growing and multiplying and harming our bodies further. But now the problem is that harmful bacteria can grow immune to sulfonamide. If bacteria grows resistant to sulfonamide, it makes it ineffective, and the bacteria is going to continue to mutate and spread. So next I am going to be talking about
Where does antibiotic resistance develop? Antibiotic resistance occurs in multiple different settings, including clinical environments like hospitals and healthcare facilities, natural environments like soil, water, and different types of ecosystems, and agriculture and wastewater systems. Bacteria is found inside of the soil, water, and different living organisms like wildlife, which are exposed to antibiotics through human waste, agricultural runoff, and industrial pollution. There is selective pressure for bacteria to develop resistance in natural environments that are polluted through natural selection. Agricultural soil bacteria develop resistance from livestock feed antibiotics or through manure spreading containing antibiotic residues. The two examples of these types of agricultural soil bacteria that develop resistance through these methods are Pseudomonas spp and Enterococcus spp. Rivers, lakes, and coastal water can become contaminated with antibiotics from wastewater treatment plants, pharmaceutical manufacturing, or agriculture, where antibiotics can enter ecosystems. There are aquatic bacteria like Escherichia coli and Vibrio spp, which acquire resistance through gene transfer in these environments. The next thing that I'm going to talk about is different kinds of antibiotic resistance mechanisms and their operation. First, there is the target modification, which involves mutations in DHPS. Bacteria mutates the FOLP gene encoding dihydropenteroate synthase, which is abbreviated to DHPS. The mutation changes the DHPase enzyme structurally, and sulfonamide can't bind to it anymore. So the DHPS continues making folic acid from PABA because the sulfonamide can't bind to it anymore, and the bacteria continues growing. This kind of mechanism is very common in streptococci and enterococci. And the operation of this mechanism is that the mutated DHPS continues functioning while blocking drug binding. The second thing that antibiotic resistance mechanism that I'm going to talk about are efflux transport pumps. There are certain species of bacteria that have efflux pumps, which help bacteria transport sulfonamide through the cytoplasm and outside of the cell. Through this process, the cell decreases drug effectiveness, and the operation of efflux transport pumps are that they're involved in active drug export. The third antibiotic resistance mechanism that I'm going to talk about is the reduced permeability of porin charges. Sulfonamides are competitive inhibitors of DHPS. Effective sulfonamides need to travel through the outer membrane of the cell, cross the periplasmic space inside of the cell, reach the cytoplasm, and bind to DHPS instead of PABA. Reduced permeability means that the bacterial cell alters its outer membrane so less antibiotics can enter the cell. Gram-negative bacteria can alter membrane proteins, which decrease membrane permeability. And the operation of the reduced permeability of porin charges is that it limits sulfonamide diffusion into the cell. The fourth antibiotic resistance mechanism that I'm going to talk about are the alternative DHPS enzymes, the SUL genes, and horizontal gene transfer. Well-known genes found inside of different microorganisms like SEL1, SEL2, and SEL3 are resistant to sulfonamide antibiotics. They produce an alternate version of DHPS with a low affinity to sulfonamides. Bacteria in the natural environment that is polluted can acquire sulfonamide resistance genes from other bacteria through horizontal gene transfer through plasmids, intergarons, and transposons. The bacteria have both different types of genes, a proper gene type and a resistant gene type. Having both types of gene plasmids decreases the effect of the sulfonamide. The operation of alternative DHPS enzyme sol genes and horizontal gene transfer is the creation and use of the drug-resistant DHPS plasmid copy. Fifth antibiotic-resistant mechanism that I'm going to talk about is increased PBA production. You might be wondering what PBA is. It's para-aminobenzoic acid. Bacteria genetically or metabolically increase PBA production, and PBA is a critical substrate for DHPS binding to it. This is a precursor of creating folic acid and is related to the previous process that I was talking about, the first antibiotic resistance mechanism, target modification and mutation in DHPS. More PABA means that there's more potential compound to bind to the DHPS gene, which outcompetes the sulfonamide. This is common in E. coli and Staphylococcus aureus. The operation of increased PBA production is the metabolic overproduction, which floods out the antibiotic. The sixth type of antibiotic resistance mechanism that I'm going to talk about is biofilm formation. A biofilm is a protective extracellular polymeric substance abbreviated to EPS matrix that is formed around dense bacterial clusters and usually occurs in natural environments. It is the matrix made out of polysaccharides, proteins, and DNA, which act as a shield, making it impossible for antibiotics, including sulfonamides, to break inside and eliminate the bacteria. Common bacteria that create biofilms are Pseudomonas aeruginosa, and the operation of the biofilm formation antibiotic resistance mechanism is to completely ignore the antibiotics.
Following that note, we must consider the implications of bacteria resistant to antibiotics. Now, as a result of bacteria utilizing defenses, as stated before, like the efflux transport pumps or the accumulation of biofilm and other tactics, sulfonamides and other earlier forms of antibiotics become less effective. This creates a dilemma where drugs, for example, as sulfonamides, are no longer a widely used option as future generations of bacteria have mutations, like in the DHPS enzymes, that will essentially render them almost useless. Continuing that note, to combat the ongoing resistance of bacterial strains, research is aimed at improving efficacy of sulfonamides as they play a major role in fighting infection. This also applies to their counterparts, like penicillin, lipopeptides, and other antibiotics that face the same threat and require proper restraint to avoid wide-scale resistance, as over time, bacterial strains will find defenses or ways around antibiotics. Now, there are limitations of sulfonamides and resistance in bacteria that also must be considered. For example, like other antibiotics, a large drawback to sulfonamides is a bacteria's ability to mutate and resist its influence. Especially this specific antibiotic, the sulfonamides, because it has been used since the introduction of antibiotics almost a century ago. There can also be allergic reactions to the medication, like others, which can result in the Stevens-Johnson syndrome or toxic epidermal necrolysis, which can be rare but very severe if not taken care of properly.